If we understand that DNA encodes the information on how cells need to function, how they need to build proteins, for example, we really need to understand, well, how is it physically that that information is encoded in those A's, T's, C's, and G's, right? There are only 4 nucleotides in DNA. How can you take these 4 nucleotides and allow them to code for 20 different amino acids? If one nucleotide coded for 1 amino acid, you can only code for 4 amino acids. That's not going to work, right? If these 4 nucleotides, if you took them in pairs of 2, any combination of 2 nucleotides, there would be 16 different combinations. 20 amino acids, that still is not enough to code for all 20 amino acids. So what the cells do is they actually read 3 nucleotides at a time. This is called the triplet code. The cell will read a series of non-overlapping 3 nucleotide words, and each one of those 3 nucleotide words codes for a different amino acid or the beginning or the end of the polypeptide. And these 3 nucleotide words are called codons. So the ribosome really reads codons in the mRNA. Codons exist in DNA as well as the mRNA, but remember that ribosomes read the mRNA, and so it reads the codons in this mRNA in a very specific direction, right? So the ribosome will read the mRNA from a 5' to 3' sort of direction. Remember, this is that theme that we see throughout DNA replication, transcription, translation. Things are always moving that 5' to 3' direction. And so each codon or each 3 nucleotide word is going to specify one of the 20 amino acids to be placed at a certain position along the length of a polypeptide. So let's spend some time on this slide. This slide allows us to take a look at how a cell would go through transcription and translation, using the information in the DNA to create an mRNA, and using the information in the mRNA to actually make a polypeptide. So we're going to start with the DNA first. DNA here in blue. Remember, DNA is double-stranded. One of the strands is called the template strand. The template strand actually provides the template for creating the mRNA. So when a cell says, oh, I want to transcribe this particular gene, it'll separate the 2 strands of the DNA, and it'll focus on the template strand, and we'll use it to make the mRNA. The mRNA is going to be complementary to the DNA template strand. So for example, if the cell reads here that this is an adenine in the DNA, it will add a complementary base down here in orange in the mRNA. Remember, A's typically pair with T's in DNA, but RNA doesn't have any thymines, so A's are going to pair with U's. So you'll see here a uracil has been placed in that first position. And then hopping down 1 position further, you're going to have your C here in the DNA, and that will complementarily match with a G in the mRNA. C and G, A and U, A and U, and so on and so forth until you've completed building the mRNA. So the DNA template strand is used to build the complementary mRNA strand. And you'll notice that when they're built, right, the DNA is running 3' to 5' in this direction. The complementary mRNA will also be anti-parallel. So anytime you have nucleic acids, 2 strands binding to each other in a complementary fashion, the 2 strands have to run anti-parallel to each other. Okay, and then the other DNA strand here in the bottom, this is called the coding strand. The coding strand really does not directly participate in transcription. However, it's a good kind of check yourself feature. So the coding strand should be identical to the mRNA, except that in DNA you have the thymine, and in RNA you have the uracil. So you can see here T, U, G, 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 T, U, T, U, and so on and so forth. So the mRNA should be identical to the coding DNA strand, except for that T and U difference. From here, right, transcription is complete, made its mRNA. Now it can read the codons, those 3 nucleotide words, to figure out what amino acid it's coded for by that particular codon. Now, it's very important that the cell group together the right 3-letter nucleotides. You have to have the right codons being read in the correct reading frame. All right, let me give you a simple English language example. So if I have a sentence, the big red pig. It makes a lot of sense in English. I've grouped these letters in this particular sequence into 3-letter words, right? We've got the big red pig. But what if I were to shift these letters over by 1 and I instead grouped them this way? Still 3-letter words. We would have a sentence that would read, like that, and it makes absolutely no sense in the English language. So the same thing is going to happen to cells, right? If they don't read that correct grouping of nucleotides, they're going to make a polypeptide that makes absolutely 0 sense. It's not going to be able to function, do its job within the cell. So the cell will then read those 3 nucleotide codons one at a time, and it will bring in the correct amino acid to string together the long polypeptide. So in this example here, UGG, this codon codes for tryptophan, UUU codes for phenylalanine, and so on and so forth. So you might ask, well, how does a cell know which codon codes for which amino acid? Well, there is a genetic code. The cells know when you have this 3 nucleotide combination, this is what I'm supposed to do. And scientists have gone through and they figured out what all of the 60+ codons actually code for. There are 3 categories of codons. You've got your start codon, you've got amino acid codons, and you've got your stop codon. Your start codon, this is a very special codon. There's only one of these, and it's an AUG in the mRNA. And not only does it signal the start of translation, but it also will always bring in the amino acid methionine. Then there are about 60 amino acids that will code for the other various amino acids. We call these the amino acid codons. And then the last category of important codons are the stop codons. And these stop codons, there are 3 of them, and these stop codons signal the end of translation. So if we take a look at the chart on this particular slide, this is a chart that scientists have put together to help us figure out, oh, when I have the codon CGA, what does that code for? Here's how to use it. So you'll notice that 3 of the 4 sides of this chart are color-coded a deep sort of maroon, burnt orange color. You've got your first mRNA nucleotide on the left, the second on the top, and then the third on the right-hand side. So if we're looking for what does CGA code for, we will take the first nucleotide. It's a C. We'll find a C here on this column, and we'll notice that, okay, all the C's are in this row right here. Our next nucleotide was a G, so we'll look at the top of this chart. We'll find the G's and find the intersection between C and G. We'll notice that CGA has to be in this little box right here. And then the far right-hand side, this is where you can find your last nucleotide. So our last nucleotide that we're looking for is A, CGA. We can find our codon right here, and we'll notice that this particular codon codes for the amino acid arginine. You will also see the AUG codon here, right? This, remember, is the start codon, and it codes for the amino acid methionine. And then in sort of red color here, you've got your 3 stop codons, UAA, UAG, and UGA. Notice that they just code for stop in translation. They do not actually code for a new amino acid. And the really cool thing about the genetic code is that it's nearly universal. This means that the same code is used from the simplest bacteria to the most complex eukaryotic organisms. So if you were to take a gene from one organism and stick it into another organism, that new organism could effectively transcribe and translate that gene with no problems and create the same protein that was created in the original organism that that gene was taken from. This can be used in basic research. Oftentimes we attach or we create new genes, or we take one gene from one organism, put in a different gene, and add a fluorescent tag to it so that we can see, wherever there is fluorescence in the cells, that's where this particular protein is expressed, and that helps us to figure out the function of various proteins and various genes. 2 examples here. This is a tobacco plant. Tobacco doesn't usually glow, but this one is expressing a firefly gene, and you can see it glowing there. This is a pig, and this pig has had a jellyfish gene added to its genome. Pigs also normally do not glow, and yet you can see that the hooves here and the snout of this little piggy are actually glowing because they're expressing the product of this particular jellyfish gene. Jellyfish, some of them can also, they have the ability to glow. Another maybe more useful way to show how important the universal genetic code is, apart from creating cool glowing organisms, we've exploited the fact that the genetic code is essentially universal in creating medicines for humans. So for example, individuals with type 1 diabetes, they cannot produce their own insulin. They depend on insulin injections to be able to control their blood sugar so that they can continue living. The insulin that we bottle and sell as a medicine for type 1 diabetics can be produced in lower level organisms like yeast and bacteria. We can put the human insulin gene into these organisms. The organisms will make a whole bunch of that insulin protein. We can purify it, and then we can use it for medicinal purposes in humans.